For a long time, medical science treated the intrauterine environment as a dark, silent void where the fetus floated in a state of suspended animation. But modern imaging reveals a period of high-intensity biological construction. Early fetal movements were historically dismissed as pure biological reflexes, muscle twitches completely disconnected from sensory perception or cognitive processing. That model was dismantled by the introduction of functional MRI and fetal magnetoencephalography. These tools allow for the live functional assessment of the brain in utero. Because more than 99% of the human neocortex forms prenatally, the foundational hardware of human cognition is largely assembled before birth. We view fetal behavior as a direct, observable readout of central nervous system maturation. Clinicians now use 4D ultrasonography to capture these complex behavioral outputs in real time, using physical movement to gauge neurological health. By mapping exact structural development milestones to these specific physical actions, the womb functions as a precise diagnostic window. The software of fetal behavior corresponds to the hardware of brain anatomy, allowing us to map the development of the mind chronologically. The chronological map begins at 7 gestational weeks, when the first synapses form within the spinal cord. This enables the first behavioral output, vermicular or worm-like movements. These early motions are driven entirely by spinal motor neurons. This schematic illustrates a critical distinction between a localized, closed-loop spinal reflex arc and an incomplete sensory pathway ascending to the brain. At 7 weeks, a fetus will reflexively withdraw from touch. This is a spinal loop. The sensory signal remains below the level of the brain, meaning there is no cortical perception of pain at this stage. By weeks 8 and 9, general movements emerge as the medulla oblongata comes online to orchestrate multi-limb coordination. This confirms motor output precedes consciousness. The biological system builds its basic physical wiring before the central processor is capable of engaging with the signals. Between weeks 15 and 20, the subplate zone forms. This serves as the primary site for early synaptogenesis. During this window, the corpus callosum is fashioned. As indicated by the red arrow, it serves as the major interhemispheric commissure, integrating sensory and motor functions from both sides. Structural anomalies in this bridge disrupt this transfer, correlating directly with postnatal learning and motor impairments. By weeks 24 to 27, the brain establishes thalamocortical connections. This bridge allows sensory data from peripheral nociceptors to reach the somatosensory cortex for the first time. Many researchers define this anatomical milestone as the minimum physiological threshold acquired for conscious sensory perception, including the experience of pain. Reaching this threshold marks the transition from purely reflexive spinal activity to the processing of sensory inputs at the level of the cerebral cortex. Once the higher brain structures engage, we observe a shift from random motor firing toward intentional, planned actions. By week 22, fetuses demonstrate goal-oriented kinematic patterns. This hand-to-face alignment illustrates complex motor planning, where the speed and trajectory of the hand adjust based on the target. We also see evidence of active learning through a process known as habituation. By week 22, fetuses exhibit a decrease in physical response to a repeated, harmless stimulus, effectively filtering out data that is no longer salient. We know this represents active neuroplasticity rather than simple receptor fatigue, because the fetus will instantly react if a different stimulus is introduced. This immediate recovery proves the brain is making a choice about which data deserves its attention. At week 34, we see exposure learning. The fetus distinctly processes, retains, and retrieves familiar auditory stimuli, such as the mother's voice. These goal-oriented movements, combined with the capacity for habituation and memory, indicate that the prenatal brain is actively running complex software to map its environment. This diagnostic model allows neurologists and obstetricians to identify exactly when this developmental sequence fails. Because prenatal behavior maps to anatomical structure, an abnormal behavioral output serves as a reliable indicator of a central hardware failure. The CANET scoring system was developed to quantify these behaviors, providing a standardized scale for high-risk pregnancies. One primary biomarker is the neurological thumb, a thumb persistently trapped inside a clenched fist. This specific limb posture indicates damage to the upper motor system, the cerebral hemispheres, and basal ganglia, frequently resulting from severe intrauterine hypoxia. Identifying these precise behavioral aberrations allows clinicians to diagnose profound neurological impairment long before delivery. There is one final variable in fetal cognition: environmental adaptation, or how the fetus responds to intrauterine stress like malnutrition or growth restriction. The fetus operates an independent neuroendocrine axis, flooding its own system with cortisol and CRH when its environment becomes hostile. This triggers developmental programming. The cortisol forces an accelerated maturation of the lungs to ensure the infant can survive an immediate birth. However, this survival mechanism exacts a structural toll. The accelerated timeline decreases the neuronal count and synaptic density in the hippocampus. This structural loss correlates directly with postnatal vulnerabilities, including impaired learning, attention deficits, and affective disorders. Intrauterine stress alters the foundational architecture of the developing brain, prioritizing immediate survival over the complex structures needed for long-term cognition. The human brain does not power on at birth as a blank slate. Through advanced structural and functional imaging technologies, we can observe the anatomical hardware and behavioral software assemble synchronously. Spinal reflexes, intentional movements, sensory habituation, and stress adaptations are all measurable milestones of the prenatal journey. Fetal behavior provides our most powerful and direct diagnostic window into the early neurological integrity of human life.